I'm delighted to share with the membership of ISFAL, in fact, the concepts that have driven our laboratory to look for lipid mediator responses in conditions that recapitulate diseases of the retina or of the brain. The fundamental concept behind the experiments is actually to look at endogenous, intrinsic responses of either the retina or the brain when disease is imminent. Our thinking is that how is it that, for example, familiar forms of Alzheimer's disease that have the mutations, of course, from birth, clinically, the disease only appears many years later, decades later. The same is true in retinal degenerative diseases which have mutations in genes that affect vision. Yet, blindness appears many years later. What had happened during that latency period when no disease appeared, in spite of the fact that the mutations are there? And our idea is that the retina and the brain had to have mechanisms to promote survival and integrity of function under those conditions. And there had to be molecular principles that actually foster survival of the cells under those conditions. Very likely, similarly, mechanisms like the ones that I'm describing may operate in many other non-genetic forms of diseases, such as sporadic Alzheimer's disease, or also at the onset of stroke, let's say, or other pathological conditions to the brain. So we began exploring these mechanisms, and we have stumbled specifically on the omega-3 fatty acids derived from docosahexaenoic acid. Initially, the main surprise was that during the reperfusion phase of ischemia reperfusion, a model of stroke, a stereospecific molecule was produced that because of the configuration was not a free radical mediated product. It was made by an enzyme. And looking into this, we found neuroprotectin D1. And then when neuroprotectin D1 was generated in enough quantity to be able to be put back in the brain, that molecule counteracted the effects of ischemia reperfusion damage. So that was a proof of principle concept that in the central nervous system, there are built-in mechanisms of defense that involve bioactive lipid mediators. And because neuroprotectin D1 is derived from docosahexaenoic acid, several years ago, we introduced the name docosanoids for that group of molecules. So bioactive lipids play a major role in the central nervous system, particularly in the retina and in the brain, when homeostasis is being disrupted as an early response to try to maintain the integrity, the cellular integrity, the functional integrity of the tissue. So the understanding of the lipid mediators and the signaling and how they work, how they operate, is bringing us into a new view of the biology of the cells and of the nervous system in general. And there is a lot more to do in this field.